So thanks everybody for being here today. I want to say a great thanks to the organizers of our writing group. This was the writing group was really Beth Manley's baby years ago, semesters ago, and she's really she does a fabulous job. And so I'm so grateful for CAT that she does that work and supports our scholarship and our scholarly community so well. And then those of you who are in writing group know that this year we started partnering with Tom. And so he kind of brought another side to this, and it allowed us to expand it and bring more people in to the writing group. And again, just get our publications done, get our manuscripts done. And I'm just grateful to both of them for doing that. And this is where this writing workshop was kind of born, as another way to support our writing with our heavy teaching loads and service loads at Xavier. So with that said, I'm going to turn over to Tom, who will introduce our speaker.
All right. Well, thanks, Elizabeth. And Elizabeth and Beth have done a fabulous job with the writing group. So I want to introduce our speaker today, Dr. Rafael Luna. And he is the Associate Dean of the Morrissey College of Arts and Sciences at Boston College. And he's also the director of the Gateway Scholars Program at Boston College. And I met Dr. Luna a number of years ago at Louisiana Biomedical Research Network annual retreat. And it was one of these agenda items. I think it was during dinner, actually, a dinner seminar. And I looked at it and I thought, what is this? It was some seminar called The Art of Scientific Storytelling. And I thought, is this going to be about, goodness knows what this will be about. And it turned out to be one of the best seminars and most thought-provoking seminars that I've ever listened to. And it really changed the way that I think about presenting scientific writing. And so Dr. Luna will provide insights on how researchers can incorporate principles of impactful storytelling with principles of science to craft an engaging narrative. So the key here is engaging, not the dry, nasty science papers. And to actually tell a story. And these papers, this narrative will make a difference between receiving just a few citations and delivering a stimulating, thought-provoking article that makes lasting impact in the field. So if you follow his recipe, your papers are just going to be hits for everybody. And so with that, I will pass it over to Dr. Luna. And you should be able to share your screen and take it away from there.
Okay. Thank you for that introduction. That very, very kind introduction. Thank you. And I'm a bit humbled by it. So thank you for your kind words, Tom. And the feeling's mutual as far as, what you're doing at Xavier and the work that, that you and your faculty are doing there as well. So it's mutual respect there. And so when I'm presenting, I won't be able to see the chat, but if you have some questions, feel free to blur it out. I think we'll probably go about 50 minutes and leave some time for questions. I'll try to make sure we finish within the hour so that we can have time for questions, because I think in the questions would be really helpful for you. And then also that I want to make sure, you get what you came out, what you wanted. So utilizing the art of scientific storytelling to publish your research. So the game has changed, and it seems like the workforce is more competitive. And I wonder if in that, especially in biomedical research, if we can reinvent ourselves as better storytellers. We're already storytellers, but how do we get better? So I published this book in 2013. And I did that because I kept getting more requests to, share the method I was using of narrative storytelling. And when I was a postdoc at Harvard Med, and other people said, well, how are you doing that? Can you show me? And I kept getting more requests to give talks. And then when I gave a talk in Norway, a professor there says, well, where's your book? Because when we do our writing workshop, we want to make sure that we use your book and follow your guidelines. And that was quite humbling. And I didn't have a book then. And so I flew from Bergen, Norway to Reykjavik in Iceland. I was like, wow, I probably should get a book. How do I do that? And then from Reykjavik to Boston, I just pulled out my laptop and that 6-hour flight. I just started typing and putting everything together and came up with this book. And I, and God bless all my friends and family and my lab mates. They all bought the first books, but before you know it, complete strangers started buying the book, and it started really selling a lot on Amazon. And someone even texted me a picture of someone on the subway in Boston reading the book, and she took a picture of it and of that person, a complete stranger. And I was like, wow, it's really getting out there. So it was quite humbling for that as well. So we can just talk about, like, what is a story? And since a lot of you are already faculty, we kind of moved through this talk, but in a way that's helpful to you. So when we think of a story, think of conflict resolution, logical series of events, and centered on a protagonist. And that protagonist, that main character, is changed forever. Similar to like our research, if we're studying our main character would be that molecule or that drug, that pathway that's important. And so if we compare that to what's a hypothesis, a fifth grade definition would be an educated guess. And then while I was at Harvard Med, and I was putting this together, I was using this method actually when I was an undergrad at Southern University, where I was struggling, and I ended up going from failing most of my courses to doing really well in biology. I started putting things into narratives. And so that was the genesis while I was at Southern University. And I did that for my coursework when I was at LSU for my Ph.D. And I did it at Harvard Med. But then when I was combining, I had a question from someone at Harvard Med, one of the deans, is how do you combine research with storytelling? And I just kind of explained to her that it's just hypothesis validation. And you juxtapose that with conflict resolution, which basically solving for the unknown. And if we impose this the dramatic arc along a energy of activation curve for you chemists or biochemists, we'll have the hypothesis. We start off there, and then increase in tension with the energy barrier with experiments. Focus on that main character, 1, 2, and 3. Reach the highest tension point or the energy barrier, and then there's validation, and your hypothesis is resolved. So to get into the nitty gritty, if we have the order of operations for research manuscripts, how do we get started? And this is where I typically wait. So feel free to shout out, since I can't see the chat. How do you start writing when you're staring at that blank computer screen? Or some of you may use pen and paper. I still do that at times when I draft things. So what do you do when you're staring at that blank page or that blank computer screen?
I usually start with like a keyword outline, like just words, just get some words, get them down there, see the flow. And I start with the easy words first, like I want to thank this person, I want to do this, and I want to then get the juices going, something on the page, and then it gets easier.
All right, so we get some keywords. Okay, that's great. And to you and all the everyone else, like where do you get started when do you start with the title, the abstract, the results, the method, introduction?
Dr. Lynn, in the chat, people are saying, and I'm happy to serve as your voice of the chat.
I see it now. I pulled it up. Now. I start seeing the
people are saying outlines, methods. Yeah.
Okay, perfect. Perfect. Yeah, methods, outline, write out a sentence or two for the first main idea. Oh, okay, that's great. And I think there's no right or wrong way. I can just share the method that I use, which is probably similar to Jay Lakshmi. A response is like to write a sentence or two first of the main idea. So I normally do that within the title. Then I typically go to the abstract, figures, results, discussion. And the methods, I think they're in there somewhere at the end, or I know that I can always pull that out of my lab notebooks. But an outline is basically, that's also a good point as well. So there's no really right or wrong way. But what I typically do is I do that first title, and knowing that I can update it like 30, 40, or 50 times. The abstract I just write, and I kind of use that as a verbal outline, and knowing that I'll probably edit that 20 times. The figures, I've already collected some. I already know. Maybe I didn't optimize or do everything in Photoshop, but or Adobe Illustrator, but I know what the data is, and then I might have to like redo some experiments, and I do put the figures there, then results and discussion. But wherever you start, it doesn't really matter, even if because I know some people start with the introduction, but if that's okay too, because if you start with the introduction, it's like a iterative loop. It'll just keep going around and around. So you'll still have to edit it either way. So I just like editing less words than editing like a large introduction or discussion. Organized matter. So I see Thomas, because it will be gone forever, but then go back and develop an outline. Yeah, so that's a great way too. So like, what I've been hearing is that people put stuff down and then you try to fashion that afterwards. That's great. But the most important thing is to get started. So when you say you do that 1 or 2 sentences that Jay mentioned, how do we compose a title? If, even if you do it at the end of the paper, after you've written everything, how do you write a title? And you can put in the chat. I can see in the chat now. I pulled up the box, so. Or you can speak out.
Titles are the hardest part. I am so bad at titles. I love to hear what other people do.
Well, also, when I think of Dr. Luna's seminars, I think of this title thing is very important, because you want the title to tell the reader what this is about. And I'm so sick of finding references that I find on my current content search, and I've got to read the abstract to figure out what's going on. And the title is actually very misleading in many cases. Have you ever noticed that? The title often doesn't really even say what's going on, or it'll be something vague like studies in such and such, or assays of this, or measurement of this. And it's like, who cares? What did the measurement tell you?
Exactly right, Tom. And it is really difficult to write the title. That's not easy, and you rely on my co-authors for a title. I like that, Mike. That's, you can punt that over. That's great. And I like Amy, independent variable, dependent variable, population, plus some action word. So Amy, I encourage you and others to, like, if you do work with independent and dependent variables, and that's population research, I encourage you to look, as I continue going, see how these list of characters fit within your framework, and then you might be able to gravitate a little easier to this method. What was done with not too much specifics. So Felicia also said good formula, Amy. So typically I think of it as a conclusion, or maybe like a summary. What's the big thing? How do you distill the 7 years of work into one sentence? And the way, and typically in narratives, like if you distill a story to its lowest common denominator of a traditional story with a traditional story arc, nothing that, there are some that do non-traditional stories, and that's fine. But typically you would have a protagonist, main character, you have an antagonist, and then you have a conflict, and then you have a scene, resolution, and stakes. So that's the basis of a story. But if you overlap that with science, maybe the protagonist, your main character, what are you studying? What is the focus of your research? And the antagonist, how are you learning? What are you using? If you're studying a protein or a molecule, the antagonist might be something that helps you understand the protagonist. And you want to think that the protagonist is what you're going to learn about, while the antagonists are what stays the same. So it's the conflict is the problem, the scene, where is it happening? Inside the cell, nucleus, cell membrane, resolutions, the action word, like Amy mentioned, and stakes, just the research impact. So here is mine. We did the C-terminal domain of EI5, promotes star codon recognition by its dynamic interplay with EI1, 2 beta. This paper was initially rejected at Cell, and I realized why afterwards, because when it was rejected, my first co-author, Dr. Hari Babu Arthanari, mentioned that it's like, just put the paper into a story. We had biochemists, chemists, two yeast biologists, and everyone spoke different languages. It's a large collaboration. And so we had 4 different labs. And so it seemed that every lab was writing their section in a different language. So, and so when it was rejected, Harry Barathanari, who's now a professor at Harvard Med, said, Raf, just put it into a story like you've done since Southern University, since you were in Baton Rouge. I've told them all the stories of Louisiana, the great food, the fun, the music. So for 7 years, we worked together, and I just always tell wonderful stories of Louisiana. And so if you're ever interested in inviting a biochemist, he would love to go because in person, because I don't think he gets too many invites to Louisiana, but he would love to go. And so here is the C-terminal domain of EI5, the protagonist. Promotes the resolution action word, like Amy mentioned. EIF1, 2 beta are the 2 antagonists. They stay the same. We don't learn much about them. We're learning about EIF5. Dynamic interplay is the conflict. Start codon recognition is the scene that shows you that's on the 40S ribosomal subunit. If you're a biochemist, you probably already know that start codon recognition would mean that. And then also the stakes, because these protein interactions allow impacting the recognition of start codons. So that's also very important. So if we have this hypothesis, EI5 promotes start codon recognition by dynamic interplay with the EIF1 and 2 beta. We interact EI5 with the EIF1 and has an interface on EI5. Then the tension 2 is we interact EI5, our protagonist, with EIF2 beta and sees an overlapping phase. Which one's more important? And then we do ITC on tension 3. Let me just go to it. So on here, we do ITC on tension 3. This is the same dramatic arc, energy of activation, tension 1 and 2. Tension 3 would be binding affinities using ITC. And we find out that EI5 and 2 beta binds well in this heat release. So that's a stronger interaction because EI5 and EI1, there's not a strong enough reaction to be able to get a binding curve. So that lets us know the 5 and 2 beta is the better interaction. And then we have a yeast biochemistry soup here. And we do EIF1A dissociation where we have the ribosome, messenger RNA, EIF1, 1A, EIF2, and fluorescein labeled EIF1A, 1 out of 35. And so when the head of the ribosome closes, and for start codon recognition, it's like a neck. Ribosome is like a head and a neck. And the Adam's apple is where the start codon meets. And so when the mRNA comes like a scarf into the right AUG codon, the head will shut down, and it will trap this EIF1A and EIF1. 1A, it'll trap the EIF1A, and the EIF1 is released. But if there's some protein disruption, like if we do mutate the binding area of 5 and 2 beta and 5 and 1, then what happens there is that the head doesn't close and it stays open. So the normal wild type is a certain dissociation curve. And then when you do a UUG, switch up it's a faster dissociation. But if you do point mutagenesis of the interface, then the head just stays open longer. So based off this is the highest tension in our research. Then we knew that we can predict that if we have a suppressor mutation in yeast with the EIF2 beta, it's called a SUI3-2 mutation that allows the misreading at the UUG. It actually, when you do mutations, it can suppress that mutation. And we were able to predict that here based off this results. So this is our validation experiment. But then they didn't stop there. My collaborator NIH said, well, this is great, but if this really works, then EIF1 dissociates, then we have a mutation that dissociates even faster off here and the head closes. So we should be able to correct the misreading there. And instead of having a UUG, reading UUG to the AUG ratio, that it'll actually correct it. And that's what we see with our point mutagenesis, that it corrects that point mutagenesis as well. And so we were able to predict that and then bring this back to the scene on the decoding box and model it, and we're able to publish our paper. And so reports 2012. And then I went to Norway to present, and at the end of in the summer of 2012, and that's how I started writing more of this. And then 2014, we did publish this method in Nature Immunology, if you're interested in that article as well. And also, when Tom mentioned that it'll make it engaging as well. There's something about learning, and if people are taking the time to read your paper, like the 2 or 3 hours, or sometimes longer, depending on your research, or evening, or spend a day reading it, and wrestling with your paper, looking at the results. If it's fashioned into a story, it's more engaging, and they're able to extract more information out of that, which would be great for you, so that they can cite your paper as well. And typically, I would ask that when I work with faculty or others that are framing the story, whatever they're doing, I always tell them to start with the hypothesis and abstract, or at least by the time they show it to me, that they would have a hypothesis and abstract, because then it'll stop it from being a headless monster. And the reason I got this headless monster concept, I had a really good friend who I was working with, and he had a paper. He was a professor at UMass Boston, and he had a paper. So, okay, Raf, come give a talk about storytelling. And he has the book. He passed out all the book to his students and other faculty at UMass Boston when he was there. Now he's at Iowa State in the Material Science Engineering department. But he says, Raf, here's this paper. Let's put this into a story. So I looked at it and I say, well, I don't see a title and I don't see an abstract. He says, yeah, let's do that afterwards. And I said, well, didn't you have the book? Read the book? He's like, yeah, I have it right here on my desk. And I'm sitting in his office. And I say, well, did you, but in there it says in chapters 1 and 2, it kind of really tells you to start with the hypothesis and abstract. Oh, I started on the section with research, so chapter 3 or 4, something like that. I said, well, you might want to start in the beginning. And then he did that, and then was able to get his paper published. So we just give a quick example here, just to see how some of this works. There's this messenger RNAs have an M7 GTP cap with they form secondary structures in the 5' untranslated region, UTR. And there's a start codon, there's a poly A tail. And so in order for this to get recruited to the ribosome, then it binds to EI4E is a protein that binds to the cap, and then binds to this EI4F complex and recruits that, brings that to the ribosome for that head to close and on that start codon. And there's a natural inhibitor that's called the 4EBP, the EI4E binding protein. And that naturally disrupts, it's a protein that naturally disrupts EI4 from 4G. So in this paper in 2007, Cell, they found a drug, an EI4E4G inhibitor, to disrupt this interaction, and because they want to slow down protein synthesis. And this is how the drug looks. And here the benzene rings as well, with the moieties attached to it. So now I ask you, you can put in the chat, now that I can see the chat. Here's the EI4E in gray. Here's the 4EGI1 drug in the space-filled molecule that pops up the bubbles right here. And for EI4G is the magenta. It's the ribbon that, remember, that protein gets displaced upon binding either 4EBP or the EI4, the 4GI1. And here's the M7 GTP cap that binds to the EI4E. So my question to you is, who's the protagonist? And there may not be a right or wrong answer. There was one answer that my professor at Harvard Med, my postdoc advisor, had, and it was one answer, and I had another answer. So you either would subscribe to the storytelling notion, which I subscribe to, or you would be just as smart as a Harvard professor with 500 publications. So there's no right or wrong answers, but I'm curious. So who's the protagonist? Is it in gray, the EI4E, or is it the drug 4EGI1? And if you do put it, why? And we'll give you a minute. Anyone in the chat?
I'm typing.
Oh, okay, perfect.
I'm getting there.
Yeah, and I do get, there's no right or wrong answer. And so 4-AGI1 because it does the action. Oh, Tom, okay. That's the drug, okay. Tom says it's right here, the space field model. Oh, that's hard because it's so out of my field, but the drug because it does the good. Okay, Elizabeth. Anyone else? Or do we agree with Tom and Elizabeth?
I am so out of my depths here, but I was gonna say the thing that gets acted upon, forgive my total ignorance, I'm a historian.
Oh, no worries. This works.
The thing that changes, right?
Absolutely. Who's that? Elizabeth?
Yeah, Beth.
Beth. Okay, yes, absolutely. So EI4 E because this is what is acted upon. Okay. So let's walk that back, Beth. You brought up something. You said because it changes, right? And so there was a little clue that I dropped in the beginning where I mentioned that the protagonist changes and the antagonist stayed the same. But I know sometimes as biochemists, we can think the antagonist is something that does something and that antagonizes the antagonist and the agonist. And especially with pharmacy and pharmaceutical and biochemistry and medicinal chemistry, you guys could look at that quite differently. But maybe let's take Beth's historian perspective, which I would subscribe to, that if it's changing, so we see here, let's look at it. How does it change? 4E's here. And then when the drug binds, or this is a loop here. Let's see when the drug binds. Oh, then it extends its helix and it displaces the 4G, right? But we don't see any changes in 4-AGI1. And so I asked my Harvard professor, I said, I give a talk there and it didn't go so well because, I was talking to all biophysicists. A lot of them were from European biophysicists from all over the world. And it was quite new for them putting this storytelling narrative arc, right? So I had a lot of questions and we're talking about hypothesis and it went to philosophy, went to other things but if we took, Beth said the 4E changes the protein, but the drug is here and has these benzene rings, right? And so my faculty member, after my Professor Wagner took me into his office after that talk that didn't go so well. And I was like, oh, great. He asked me to give the talk to everyone because he says, you're flying to Norway, you're going all over Europe and US, giving this talk, I want you to share it with the group. But that sharing with the group didn't go so well. So then he takes me to his office and says, okay, Mr. Storyteller, who is the protagonist here? No, is it the 4-AGI1 drug or is the protein? And I said, I don't know. I was kind of like Beth, it's out of my element. I don't work on that paper. I have no idea. And then he says, and I said, well, I asked him one question. I said, so what do you think? He said, I think it's the drug. I said, why do you say that? He says, because it should be the drug. And then I said, okay, well, I want you to remember how this picture looks. There's the benzene rings and these moieties attached to it. But then I asked him, I said, well, does the drug change when it binds to the protein? He says, no, those benzene rings. I said, do they open up? Does the molecule break in half? Does it multimerize? Those side molecule moieties, do they multimerize with another molecule, make a bigger form aggregate? Does the drug do that? He said, nope, doesn't do anything. We look by NMR 1D and you can see that nothing happened to the drug at all. It doesn't change at all. I said, okay, well, that tells me it's an antagonist. So then I said, what about the protein? Is there a change there? And he says, well, actually, there's an extension of a helix and it displaces allosteric conformation. We know that, but we're having this hard time publishing this paper. So I said, oh, so that's obviously then it's the protein. He says, no, it has to be the drug. And I was just like, okay. But then I said, well, wasn't your drug the protagonist in the Cell paper 2007? This is like 2013. I said, if the drug was the protagonist there, you characterized it, you did the IC50, you did everything, and you characterized the drug and saw its infectiousness in Jurkat cells. So that was already you already told that story. So here, you're telling the story of 4E and how it changes. So yeah, so Beth, you from the storytelling perspective, it was you're correct from there's the 4E, but you can still write a paper from the 4E GI1 perspective. Now, but then it'll be a harder narrative to feel, right? Because if you're watching something like on Netflix, Amazon, you want to see a change in that protagonist. Um, and you want to see and to learn something. And so it would be harder, and I can just tell from Professor Wagner's experience, he had 16 papers. They were working on this for 5 or 6 years. They couldn't get this paper done. They had all the results. They had the crystal structure, and they still weren't able to publish this paper. It didn't congeal. And for 450 papers at the time, he would normally go from the results and build up from the results to the results. I mean, the figures, results, and then build out and to have your paper. And that worked great for him for 450 papers. But it was stuck here because he kept doing based off the figures, but then he just kept adding more figures, like 17 figures he had, and he just kept collecting figures. But part of it, if you looked at it from the protagonist perspective from 4E, then it was quite easy. And so we're able to publish the paper, and there's no good deed that goes undone. So I was able to get published on that paper. But in order for us to get published, we did mutagenesis all over 4E to really validate that that the correct binding site is the right place. Yes, so the med chemist is focused on the drug that they spend so much time optimizing. The reader wants to know how the drug is impacted. Yes. Good point, Tom. Great point. And you know, and I think sometimes that brings a good point. So when we talk about antagonist, protagonist, you know, and it's not that we're saying evil or good villain or villain, like as if you watch something on, you know, like a horror movie or a comic book, Marvel comics or the Avengers. It's not like that. It's science. So for us, we're not invested in the outcome as far as we want our protein to do this. We follow the data, right? So if we look at it and remove any part of like good or bad and just want to understand how these two interact, then you're looking at what changes and what doesn't, and how to really tell that story. So we're able to publish this paper, and they were stuck on this for over 2 years before asking me, and then we were able to, in like 2.5 months, publish this paper. Do you know that there were other labs across the world that were working on the same thing, because they had at the same time, and we beat them by a month on one paper that probably would have gone to cell, but it ended up going to molecules and cells because we scooped them. We beat them to the punch, and then they published the models and cells. And then there was like another paper. I don't know if it was like a science journal that they used, or another PNS paper. And they all had to cite our paper because we were there first. And so it does make a difference. If it can get you to publish that paper quicker and engaging, it's really helpful. And herpes virus is like true love. It lasts forever. And we just want to kind of go through these slides a little bit quickly. That we have herpes virus inside the nucleus, and it's double-stranded DNA. And here's all red is where the capsules are filled with the nucleus, and then it kind of busts the internuclear membrane, buds out into forms a vesicle, and goes out to the outer nuclear membrane, and eventually cell lysis, and the virus spreads and viral egress. And so human cytomegalovirus is a herpes virus as well. And then you have UL50. UL50 is a transmembrane protein, has a transmembrane region, and that allows it to anchor in the transmembrane. And UL53 does not have a transmembrane region, and so it can only line the membrane if it's bound to UL50. And then so in mouse cytomegalovirus, this MCV, the M53, is the same as humans, but it just easier, behaves better for protein biochemistry reasons. So if you look at this M50, it's a form of a beta taco that's in the paper. And here's the M50 with a transmembrane region at the top. And you have M53 that can't find the nuclear membrane without binding to M50. And then here in the red is where M53 binds to M50. But if you do point mutagenesis, then you can get a binding curve in wild type. And then if you do point mutagenesis, you could disrupt that. And so they knew that they had the binding interface. And so what happens if you not disrupt this interaction? So normally if you have flag tag UO53, and UO53, remember, binds to UO50. And so this flag tag you see in red, it'll line the nuclear membrane. Here's DAPI stain, and you see the merge. But now if you knock out the gene of UO50 that brings UO53 to the membrane, you see it dispersed throughout the whole nucleus. But if you do a point mutagenesis, the right region of that interface, and then UO50, then UO53 would be diffused, just as if a point mutagenesis would be the same as knocking out the whole gene. It'll diffuse out. And so that was also the validating experience. And then you can see here that the paper was published. And this is a great title. This is Hari Barbarathanari that encouraged me to publish our first co-author paper on that EI5 protein. And so he's doing this paper. He's the corresponding author. And he says, great, I followed your method. I said, well, that's great, but your title's kind of long. I know you have all these elements. And so here I said, well, you don't need the word that is. This reveals an interaction group essential for viral replication. So I think that was enough. And so, just maybe you could have deleted those words. And it was too late. I said, the paper's published, you're right, it's too late. However, I said, don't worry, Hari. When I go give my talks, I'll always mention your paper. Say, no, don't do that. It's going to be embarrassing to me. I said, oh, no, don't worry. I'll make sure to cite your paper as well. So I have his citation here as well for PNAS, and make sure you get all the credit. And he didn't like that very much, but he's my buddy. And so we kind of joke with each other a lot. And here's a paper that had nothing to do with when I gave a talk in Austria. It's a beautiful story. So here, can anyone identify the protagonist? Anyone see the protagonist? Can anyone identify the conflict, the scene, the resolution? It's a beautiful story, this paper. And they submitted for it to be workshop, but he submitted the preprint for PNAS. I'm like, well, I don't know how I'm gonna edit this or help you because it's already going to be published. But he says, I wanted your perspective because I had a hard time publishing this paper. I said, okay. And I read the paper, but it was so beautiful. But when I read it from my scientific lens, it was hard for me to understand how to give it a critique to storytelling. But I was jet lagged at 2 a.m. I flew to Europe and stayed up late, and then had jet lag and had to talk early in the morning. I should have gone a day or two earlier to adjust, and I could not. His paper was the one I started with, and I put it at the end, and I was at 2 a.m. I was like, I've got to figure out how to provide critique to Georg, and I couldn't. A conflict, endotoxicity, absolutely correct. That's correct, Thomas. Anyone else, like, on where's the protagonist? But every time I looked at it from when I read it, and I was so tired, I said, there's nothing wrong with this paper. It's a beautiful paper. It's a story, right? But then, so this paper was fashioned to a story, intentionally, and it was beautifully done. Peptide is the protagonist, absolutely. Is it Elizabeth? Yes, Elizabeth. Yep, that's the peptide's great. Is a protagonist, non-ribosomal peptide. So, what is causes and what's antibiotics associated colitis? We're running out of words, and so. But I was so frustrated because I couldn't understand how to critique. I was like, is it possible that this is the perfect paper? That I finally met the holy grail of papers, that there is a perfect manuscript, which in all of my years, I haven't found one. Maybe you have read perfect papers, or maybe one of yours papers are perfect. I just haven't seen one in my work that I've done. But they may be out there. So I finally thought I came across that that white whale of a paper that this is the perfect paper. But that was when I was reading as a scientist. And so Tom mentioned, is colitis the antagonist? Does everyone agree with that? Colitis, could that be? What did colitis let you know?
Isn't that the result? What happens?
Yeah. So colitis, so kind of like the impact, right? Like the antibiotic associated. The antibiotic kind of gives it the research impact. And the colitis is kind of like the scene. So you know it's probably like some epithelial cells, endothelial cells, but germane to the colon or into the digestive area. Tom is disagreeing with himself. That's great. We could definitely do that. And so when I at 2 a.m, I was like, I don't know. I just have to finally give up and say I don't have a critique of this paper. I have critiques of all the other papers. It's the Holy Grail papers, and God bless you. You've written the perfect paper. I have no feedback. But then I said, wait a second. If I put on my storytelling lens, I did exactly this, and in 3 minutes I said, enterotoxicity, boom, conflict, non-ribosomal peptide, protagonist. This is the way I viewed it. Causes, the resolution, action word, like Amy mentioned, and but I wouldn't agree with the word causes because my professor Gerhard Wagner doesn't like strong words like that. It's you get information that leads to a result. So maybe this word could have been induces, leads to, but nonetheless, it's a resolution and fits an action verb. So that's a resolution. And then antibiotic associated colitis, that lets me know where it is. This enterotoxicity is there in the colitis, and that antibiotic associated is the stakes. That's the impact. It's not only is enterotoxic, but it's antibiotic associated. But then I say, where's the antagonist? So then in 3 minutes, I realized, oh, I know why this paper, Georg had such a hard time publishing paper. It was rejected from Cell Science and Nature. And I said, maybe I know what, I can't say I know why, but maybe this is part of the reason. Maybe it's missing the antagonist. And I looked in the abstract, couldn't find it. I looked in the results, I couldn't find the antagonist. And what does that mean biochemically if you don't have the antagonist? And for this story, now putting back your scientific lens, if you don't have your antagonist here, then what does that mean? So if you're saying not, let's look at this, break this down scientifically. Non-ribosomal peptide, enterotoxicity, if it causes antibiotic associated colitis. How do we know that that's correct? How do we know that that's correct?
Well, if you don't have a change agent, how do you have change?
Right.
Is that right?
Yeah, yes. So we know the non-ribosomal peptide. If we can look at the non-ribosomal peptide, it's similar to the bug in that first 2007 paper that they followed the peptide. They showed where they got the peptide. They showed how this peptide infects cells, enterotoxicity in cell lines, and I think even in rabbit. But if you're saying that a peptide causes enterotoxicity and colitis, how do we know? How do peptides work? Would it just do it by itself? How do we know? If it's a peptide that's causing something to be toxic, how do we know that's correct? Are we going to believe them? Are we going to just believe Georg? So, okay, thanks for, we believe you. But at Harvard Med as well, my postdoc, there was someone, my boss there, his PI for grad and his postdoc PI won the Nobel Prize, and he told someone in his lab, belief is for the church. So if we're to believe a non-ribosomal peptide causes enterotoxicity and antibiotic associated colitis, are we sure about that? And yes, Beth, I'm sorry you're lost, but if you can just bear with me, how do we know that would be true? If we just look at Lego blocks, how do we know something's true? If there's an action and there's a reaction, if we're saying this, doing this, then the first question would be, I knew once they didn't have an antagonist, the storyteller in me told me, oh, they don't have a mechanism. Because if they don't show where this peptide binds to a receptor, then how do we, and that receptor is, in the same area, it's like endothelial cells or wherever that receptor is, and leading to enterotoxicity, we don't realize that and do experiments also on the receptor, then there's no way that I could just by face believe this non-ribosomal peptide. And so I'm sorry, Beth, if this was too techie or science on this part, but the key is if you have an action or you're saying something, how do we substantiate that? Because remember, the antagonist is there only to help us illuminate what's in the protagonist. So if we put our storytelling lens on, then if we want to understand the protagonist of a non-ribosomal peptide, then we're only going to learn that through the interaction with antagonists. But if we don't have antagonists, then we don't have a mechanism. We don't really know how this works. And so that's the really key part. And so, and Beth, you're doing great, by the way. You're doing great because you did pick out the proper protagonist. So you're doing fantastic.
Thank you.
So you did fantastic. You're hanging in there. And so here, the protagonists were two protagonists, 1A and EI5. It's like Hansel and Gretel narrative. Retains the resolution. EI1 is the antagonist. And scanning pre-initiation complexes the scene and stakes. This was where a process, magnetic levitation, to separate. That was the first time a protagonist was a process that we found. That took us 4 hours to come up with that title. I thought that was, we published this in Nature Communications paper. And here, this physicist had a hard time publishing this. And then I encouraged him to put an action word like Amy mentioned and quantifies to kind of bring everything together. He was able to publish this paper. And here's Martin Thuo, the one with the headless monster. He came up with Slice, but before he didn't have a name for the process. I said, well, your protagonist doesn't have a name, and so you need to come up with a name. He says, no, somebody's going to cite me, and they're going to come up with a name. I said, nope. If you want a broader impact, you need to come up with a name. You need to name that baby. If you can't take a baby out of the hospital without a name in the baby, then you certainly shouldn't be putting your paper out without naming the process that you came up with and you worked so hard to do. And he came up with Slice, and this helped him get his position while he was to move to Iowa State as a faculty there, and he just received tenure as well. And here's someone, Sarah Musa. She's a professor at Duke now. She was a postdoc. She was publishing her graduate work. I met her as a postdoc, and she had a hard time. She used the method to publish a PNAS paper. Here's someone, Scott Loki. I never met him. He was having a hard time publishing this paper. A friend of mine that read the book said, hey, read this book. And he used the book and they was published this in JAX. And here's someone that caught me in Germany when I gave a talk at a conference, Karsten Boll. And he worked with Professor Mürklich. And I was running my daughter was living in Germany at the time. And I was running to go take a train to go see her in Germany. And he's like, no. I said, and I had 20 minutes before the train arrived. And the hotel was right outside of the train station. And I was going out the lobby. He's like, no, I need, just give me a few minutes. Let me explain my research to you because I can't come up with a title, And so I was like, I've got to go wait for this train because I got 25 minutes before it comes. He was like, that's plenty of time. Just give me 10 minutes. I said, no, I need to go wait by the train. He said, no, you don't have to do this. This is Germany. The trains run on time. I said, huh, really? He said, yeah, don't worry. It would not leave early. And so you have the time allotted. And I said, okay. So we sat in the lobby. I gave him 15 minutes and I got nervous. And we came up with the protagonist, the multi-layers, maximum energy density of the antagonist, and along with ductility, reactive materials was the scene and the stakes, and integrates the resolution. And he was able to publish this paper, and he sent the paper to me a few years later saying, hey, the paper was published with the same title that we came up with in 10 minutes at that hotel lobby. And here's Professor Musa again. Now she's at Duke, and she published her paper, this Nature Biomedical Engineering, but this is the paper that allowed her to do her job search, and she was able to land her position at Duke, and now she's publishing, and I'm so proud of her as well. And here's Professor Arthanari again. He says, okay, Rafael, here's this paper. I fixed the title. The title works now. We got the paper published in Nature. And he says, so what do you think? Does it fit all the 6 elements? Protagonist, antagonist, conflict, scene, resolution. And he sat right behind me, and he had a 1990s version of a Linux keyboard, and he would just bang on that all the time. And I said, we're in the same Bay Area of our office. And I said, this doesn't have the elements, because I don't know who's your protagonist, antagonist. I don't know, is the resolution inhibiting or disrupting? I'm a little confused. I don't really know where the scene is. Maybe fungus, like, I don't know. I said, but just to back up a little bit, Hari had an issue with the same problem. He had a drug, and so similar to the 4G one in the 2007 paper that published in Cell, he characterized this drug and found the drug and characterized it, pulled it out of the library. But it was named something like ICCB. I'm gonna make up a name like 3, 4, 5, 8, R2D2 719 ICCB, and he put his protagonist all throughout that number, that code of that molecule throughout the whole paper. I said, dude, and I already worked with Professor Thuo on his invisible protagonist that didn't have a name. I said, you got a name for your protagonist, but your name is not really working. He said, well, no, I'm a chemist and I need to stay true, and that's the molecule, and we're gonna. I said, yeah, but you could rename it in a way so that your audience can understand what it does, and then also you'll lose your audience if going to nature. It's such a general audience, you'll lose your nature with that molecule. And he says, no. Well, he says, okay, Raph, well, give me the name then. You tell me, you give me the name of that. So he tried to punt it off to me, but I wasn't his co-author. My name is nowhere on this paper. And so I said, that's great, Harry, but last time I did that, y'all put me in the lab doing all these point mutagenesis, and I'm not falling for that. Nope. You need to come up with this. You've sat behind me for 5 years, 6 years. So now it's your turn to come up with your name of your protagonist. He was so upset, banging on his Linux keyboard so loudly. And I was like, wow. And for like 3-4 weeks, he'll come up with a name. And I said, nope, that still sounds just like you're going to lose your audience. That name, you could do better. Dig deep. And then finally he asked he came up with something with the ARC mediator, med inhibitor. And then he says, wait a second, it's the Kixin domain. And Anders Nahr says, well, how about we call it the iKix? He says, Raph, what do you think about that name? I said, it's a beautiful name. Published the paper in Nature. And he used this to get his job at Harvard Med. And he was able to publish the paper. So what I learned throughout my journey is that it's not only to publish or perish, but ending the stories. In order to publish, you got to finish the stories, and narratives just help you finish that story. Just like the 4E being the protagonist, helped my professor finish that story and beat everybody else. And so he didn't have to cite them and have a PNAS paper. Or if not, his paper wouldn't have been in PNAS. It would have probably been in a less high impact journal. And so ending stories matter because it helps you get faculty positions. And so here's the Hall of Fame storytelling. Here's Professor Samira Musa, who does all the biomedical chip things on Nature Biomedical Engineering. Professor Blair, he used it for, he's an economist and working on health disparities. He did his Ph.D. At Wharton, and he used this method for his Ph.D. Professor Thuo's here. He did that with the invisible protagonist that he fixed. He's doing great. And here, my Professor Wagner, who encouraged me, and we had I learned so much about science just from him. And here are people from all over the world, Danish scientists, Israeli, Austrian, and another Austrian faculty, and Professor Asano and Asen, who are my mentors as well, and that helped me publish those papers. So I just want to thank Tom for inviting me again. And here I'll be happy to answer any questions. I try to get through and finish before 2:00, so I want to be respectful of your time.
Any questions for Dr. Luna?
Well, we have a whole hour. If people want to stay, we could use that to do Q&A. It's totally up to you, Tom. It's just we could take a couple minutes here, and then it's up to people here.
yeah, we were thinking if we can take 5 minutes here and let Dr. Luna catch his breath here. And then if you want to stick around and talk about particular scenarios with him, this is a great opportunity. I know all of you guys are working on different papers, so we can do that.
I was going to add, we can maybe ask if you've got a general questions, ask, but also specific ones for your exact studies. I have one general one before we take a break. I do think, like, let's take a break and then come back with the doing more consultation. But my general one that I've been thinking about and I haven't found the answer because, I'm a psychologist, by the way, by training. I'm a social psychologist. Is when you have a standard IV-DV studies, right? Like, so that's my standard studies have IV, human behavior, IV-DV. Is there a general protagonist, antagonist? Is what's the usual there?
That's a great question. This is a question I get a lot from like psychologists or like people that do population. I get that a lot. And since this is not my field, I kind of feel like Beth, with the science, I would hate to speak because I wouldn't know for all scenarios. Right, for biochemistry and virology, I can certainly say absolutely fits for all scenarios. As far as your protagonist, what changes and what doesn't. But what I would encourage you to think is, because sometimes maybe your independent variable or your dependent variable, it could be depending, but at times you might need to look at it, because typically in biochemistry, the drug is going to be the protagonist. Like almost 9 out of 10 times is going to be the drug. Okay, maybe even 99 out of 100 is going to be the drug, right, for a biochemist. But in this one instance, it wasn't the protein. What it was being acted upon was the protagonist. And Beth nailed that as a historian, right? And so, maybe 99 out of 10, it could be one or the other. But I would hate to say that, because if you go by that, because I think the biochemist always going to think the drug's the protagonist, in the main character. We're focusing on the drug because we need to, market that and do everything else with the drug. But it may or may not be, depending, and it depends on the study.
Yeah.
What are the other antagonists? So I would say, to answer your question, I think it would depend on the other antagonists in the narrative and your tension points. How are you evaluating this? And what are the other antagonists that you're learning from? And then based off of that, and on a specific paper, if we could discuss, then I can tell you if this is the protagonist or not.
That's kind of what I was thinking. Like, if you had an Amy are you thinking this too? I'm thinking Amy, because she does this kind of research as well. Like, so if you had mediators or moderators, it might be a different story than if you were just doing a strict IV-DV, which it would be like the IV. Okay, that's thank you. That helps a lot.
Yeah, but so I would think I get this question a lot, but I've seen so many complex studies, and the IV-DV do so differently that and even like some of the work in mentoring and statistics and national, it gets complicated. So I hate to say one for the all. So, but from the storytelling perspective, that is pretty standard from the beginning of humankind. Like, there's a protagonist that changes, and then the antagonist you learn from the protagonist. So I see Beth now. Did I get that right, Beth? Because you're a historian. This is more your realm than mine, because you're more of a storyteller than I am.
Totally, but I don't necessarily always think about my own pieces in this way, right? Even though like when you asked how you start an article, I'm like, I start with an anecdote. I start with a story. Like, that's how I start my work. But I'm like thinking about this article that I'm writing right now, and I'm like, do I have a protagonist, or do I have a thing that changes? I have a lot of things changing, but it's a really profound way of thinking about your writing.
Oh, thank you. I've seen it work. I haven't had it not work. Worked for reviews, physics, socioeconomics, economics papers. I wrote review papers, which is not really scientific, but review paper. I put that into a narrative, and so like almost anything, the letters I do for my pre-health students, I make sure there's a dramatic curve, and is it a validation for, if I'm saying the student's awesome, am I validating it? How is the student awesome so that it's correct, right? So, I try to make sure that's, so I use that as well. So, yeah, but it's a little difficult to be kind of like intentional about it. But if you're intentional, it can accelerate the writing process. It's just another tool.
But whenever I hear you present, it always brings me back to my advisor looking at things that I would put together for him, and he'd look at it and say, why do I care about this? And he'd sort of slap it down. And I think that gets to the point is you need to have a story that's identifiable. I was so obsessed with that process that it created all this data. And he's like, why do I care? I was doing all this molecular modeling of something that some process in the lab. And he's like, well, it looks pretty. I don't see why I would care about this.
Yeah, and he's probably asking the stakes. Like, what's the stakes of your story? You're doing this, but if you put the context, like, is it the antibiotic-associated colitis? Is it start codon recognition? Is it, anti-drug? What is that impact? And then you're able to really evaluate how effective that is. And then you can say, okay, if that's what you're claiming, then we can look at your model. Does that really do that? And does it really say that, right? And so it also helps the reader to frame it better. It helps the reader to be able to evaluate and judge your work, which you want to, right? You want them to judge and say, okay, well, if you submitted your paper, you want your reviewers 1, 2, especially that reviewer 3. You want that reviewer 3 to get your story, right? And you want them to give you experiments that you can do because he understands your narrative. That you want them to suggest experiments that's within your narrative, right? And so and how many times you submit a paper and they suggest experiments that's not even part of your story, like that has nothing to do with your paper, but they miss that narrative, right? And so that's why they're suggesting that. And so, but if you can kind of keep them within that story and they get it, then they can ask questions judging your work and helping you with new experiments that could get your paper accepted. I never really liked doing extra work with manuscripts, but then I realized it seemed like every paper we submitted, the reviewers always asked for more work. If not, the reviewer 3 tried to reject it, but sometimes reviewer 1 and 2 really kind of stick up for me and say, okay, no, let's see these experiments before we reject it. So, but if they get within a narrative, it's really helpful.
Also we can think about this in terms of seminars we give. When you give a seminar and then you ask for questions and the question is way out in left field, you realize that maybe you should have presented this in a way that tied up the story a little better. or even class, when you present class and then students are all fixated on something that really is not the main issue that you were trying to discuss. And they're all fixated on that. That means I should have used a different approach there.
Yeah. And then like that paper was a real story about how I figured out that narrative in 3 minutes, just based off the title. Mm-hmm. So even if you're maybe like you might be lost, like, in someone's giving all this research and they may have lost you on one of the first figures in that talk. But if you don't see that, if you have the storytelling lens, you still might be able to ask a really informative question about that research, or be helpful to that presenter with aspects just based off of the antagonist. You wouldn't say, for that, if I heard them talk, they would have presented for that non-ribosomal peptide, they would have presented how they identify the peptide, they pulled it out of cells, they show how it's where it's located on cells, they show how enterotoxicity, they showed that. But then I would have asked, oh, they need an antagonist. I don't see an antagonist. I don't see anything binding to that peptide. And so if nothing binds to that peptide, that tells me there's no mechanism. So you can ask how, what would be the mechanism of the peptide? And then that's a fair question. And you could maybe not even have, if he had that same title for the talk, you may not even have to listen to the rest of the talk. You could have either, had that question initially. And if the talk doesn't have it, then you can ask that question as well. So I use that also to ask questions. And I just like to do everything through stories. I tell my daughter I'm a one-trick pony. I just do one thing and I just do that. That's all I typically do. And it's just through stories, whether I'm talking to friends, giving advice, advising at work, it's everything through a narrative. Because they say, what's your advice, Dean Luna, if you want this or that, or our friends. I say, well, let me tell you a story and then maybe you can decide. And then they were like, great, this is going to take longer. And it typically takes longer. But yeah, I found if you give advice, most people don't necessarily take it. They do what they want. But if you present a story, you might, induce them to kind of think about
Right.
both options. So and I do that with my daughter.
Has anyone ever called you the title whisperer? Because I think you are.
Oh, that's so nice. Actually, that's a new one. That's actually kind of cool. That's kind of cool.
Yeah.
But yeah, the titles come up a lot faster now, and it makes writing fun because I used to dread paper writing, or manuscript writing, and I'm working on my second book, and so like I have to use this for myself. I'll say, what's the narrative? What am I saying? What am I including? And I have to be a little bit more thoughtful on that.
So it also occurred to me that maybe we need to, as colleagues here in the writing group or at the university in general, use people outside our discipline to read our material to see, if it's clear at all what we're trying to get across. And we could have, Beth could be the evaluator for all the biochemistry, and
that would be amazing. You will have so many insight just from questions that she can say, well, what about this and what about that, and it'll be quite.
I think it would be.
I think that'd be quite innovative, and your papers would be really getting out a lot faster. Because I think part of it is that is that in the book I've mentioned is that last experiment, figure 6 or 7, like it just takes forever, and then the more you try to do it, get the results, optimize that experiment, there's something that doesn't go right, the gel, the protein, and you kind of keep redoing it, or something new. It's like, oh, it doesn't fit, like, and you keep, and then maybe that's not even part of the story, and then you learn the narrative method helps you just get rid of that figure and just end it. If your hypothesis is substantiated, you don't need to keep hammering that home with other stuff, because that's probably a different story. Not all the time, but a lot of times that experiment, you're agonizing that last experiment, maybe that's a different paper, because you're not going to solve cancer in one paper. It just it's not going to happen. When we do have a cure for cancer, it's going to be in multiple papers from multiple labs. So do your part. It'll be too small to fail. That's what I heard recently. I said that's kind of cool. You want to be too small to fail.
Too small to fail. Interesting. Well, I think we could all fit into that.
Yeah, same here.
All right. Well, I guess we've sort of blended into this next section here. Does anybody have any stuff they want to run by Dr. Luna, or any ideas? Or we can even set it up so I think everybody can share the screen.
Do you want to introduce yourself to him first before you dive in and submit yourself to critique?
Well, I just wanted to real quick, because I've got to run myself, but I have a more generalized question that I'm more interested in. Thank you again for the talk. This is the second time I've heard you.
It was great meeting you. Yeah, back when I was visited. Yeah.
Yeah. But yeah, I guess from my perspective as a pharmacist here, I'm writing more about patient care and that type of thing. So would you say that when there's an intervention, a medication intervention that is put forward, would then the patient be the protagonist, as they're the one that's changing, and that I guess the disease state itself, so I'm in psychiatry, so schizophrenia as an example, would that be the antagonist because that is causing harm to the patient?
Right. That's a great, I remember we discussed that paper you were working on before, which I really enjoyed your work when we were talking about that in your office. Based off of what you told me, the first thing I get from the impression from the narrative would be that the patients wouldn't be the protagonist, because to me, I think it would be the intervention, right? And so if you have schizophrenia as the disease, people understand that, right? Unless you're going to add more to schizophrenia, but schizophrenia people probably have it's not phenotypes, but disease manifestations that are very clear and delineated for schizophrenia. But the intervention might be new, right? And so you probably had to present that. And so I think that would be the protagonist. And how do you validate that? How do you show that intervention works? And so maybe the people might be the antagonist, because if the intervention really works, it should work for the majority of people, right? And so if it's really effective, right? And so I would think maybe you have different population groups with different cohorts impacting an intervention. Then have you tested on this population? Have you tested on this other population? Does the intervention work? Does your model only work for certain populations, or could it be for everyone that has schizophrenia? And so I think that, doing that and testing with different populations and different surveys or experiments that you have, I think that would be, to me, that would be the protagonist, because that's something new to the field. And that's what we learn. But we're going to learn it through your antagonists, which are people. And even if it's different population demographics, those people should probably be impacted the same by that intervention, even though it's not 100%, because no intervention is going to work exactly for everyone. But overall, there'll be, it should kind of work. Does that make sense?
Yeah, no, absolutely. That puts it in great perspective, because I think, looking at the sort of drug targets and how we're aiming to various populations, you can definitely tell the story of what it's doing to each of those different populations and walk them through that. So yeah, I think that's very helpful advice there. So yeah, I definitely appreciate that.
Absolutely.
Well, great. Well, thank you so much. And I'm sorry I have to step off as
no
well.
No worries.
This was excellent.
Thanks, Thomas.
Really appreciate it. Thank you.
Thanks, Terry.
All right.
Amy, you wanted to jump in?
Yeah, you might remember me.
I
we went out to dinner.
Yeah, we had the oysters, right? Didn't we have the oysters?
We had a lot of things.
Oh my Lord, that was such a great night. Oh, that was great.
It was a good dinner. Yeah, and so I think my question kind of follows what Tom just asked about, because I'm a population health scientist, so I'm usually looking at some health outcome, as my dependent variable, and then some, often a social condition or an environmental condition, to see how that might impact the dependent variable. And so I've seen it done different ways, and people have given different opinions. But in terms of, I guess, the storytelling, one thing I always struggle making a decision about is, do I start my story talking about the health outcome and kind of describing why it's bad and there are disparities? Or do I start by saying there's, this social condition or this stressor that is more broadly, negatively impacting all kinds of health outcomes, or, influencing disparities? And so that's one thing that I always kind of struggle with, because like I said, I've seen it done both ways. People, I think, can maybe have a preference for one versus the other. But in terms of the storytelling, I don't know if you have an opinion or advice about what piece do you start with?
That's great. That's a great opinion. And if I, go into the narrative aspect, I would think it would be a point of view, right? And so maybe Beth might want to add on to this here. Where's the point of view? Like, I think when, like you say, we're just taking film. So when I have the same question, where do we start? You can start in so many different places. Like, your question's a real one, right? But just like a movie director or any story, you can start with so many different. But typically, a lot of times, like the camera, I think about the point of view. The point of view is focused on the protagonist. So you mentioned your protagonist is what was the protagonist again?
So I guess it was, like I said, kind of following up with what Thomas had asked about, because I study people, but I'm really looking at some specific condition, like a health condition or health status, as the dependent variable. So to look, in terms of, is there differences in that particular condition, based on people's exposure to some external environmental condition.
Okay. And then your antagonist would be what?
So for example, a lot of my current work is focusing on neighborhood conditions and how those influence or predict states of health.
Okay. condition? I'm just typing it out here so far. This is a good question. Neighborhood conditions that impact what?
Various health outcomes. So in one case, like looking at cognitive functioning or physical disability, as two examples.
Okay. This is good. So what I like to do is really focus on that protagonist like early on. Or bring that protagonist in after you present the problem, but it has to be quick. So if you're watching a movie, and there's like Keanu Reeves with The Matrix, I think it opens up with him, typing on the computer, he's taking the red pill, the blue pill. It's on him. He's the protagonist, right? And then there's a problem right away. So you can do that way with the protagonist, being the health condition as the dependent. You can focus on that, which pill do you take, the blue or the red? I forget how it starts. But then, or like the Lion King, where it starts with Simba being lifted up, right? And so, and that's the protagonist, very clear. But then the problem came right after in the Lion King. The problem came right after in the Matrix. And most films, if you have your protagonist, the problem comes right away. But in science, this is tricky. I think for me, like personally, I probably would appreciate the problem first, and then the antagonist coming in, and maybe. And why do I like what Tom mentioned? Why would I care? You present the problem, and then it will lead the reader that something needs to be done, or an intervention needs to be addressed here. And then this is why, informing your hypothesis, you led to this. And I think that's a fair and safe way to do it. But I've seen both where you start with the protagonist, but I think here I would start with the antagonist or the problem, the problem and the stakes. Certainly, but you've got to get to that protagonist early on, that first paragraph. At the end of that first paragraph of the introduction, it has to be in there. And in the first, second, or third line, the protagonist needs to be in there. But typically, we start very general, and then we go into it. And then after you introduce the protagonist, then you say, what is that protagonist going to do? How is Keanu Reeves going to, he gets pulled out of the Matrix, how is he going to do this, right? And then you go into the rest of your paper and all your experimentation. And then at the end, you want to go back to the end, where remember at the end of Matrix, it's focused on Keanu Reeves, he flies away, right? And so it goes back to that protagonist. But then at the end, maybe you go back to the problem, not necessarily the antagonist, but the bigger problem, but how it's solved and how what you're suggesting might be a better, how do you put your research in context of that? Does that make sense?
It does. No, yeah, that's really helpful. Perspective, just sort of a way to frame it, because like I said, that's many times when I sit down to write, like, okay, well, what do I want to start? And it is, I have kind of a narrative in mind, but it's like, how do I want to get people hooked in because of my dependent variable, because they care about Alzheimer's disease, or do I want to hook them in to introduce this maybe novel or interesting independent variable or exposure. So kind of thinking about it in the way that you proposed, I think that makes sense for my work. I appreciate it.
Yeah, and at the end of the day, if you want to do it some other way, that's fine too. But I think the biggest thing is to submit it and then get feedback, and then people say, well, does this make sense? Or then I like writing everything, even if it's a horrible first draft, and just editing it afterwards, so I wouldn't stall. I would just make sure to just keep moving along. So I hope that's helpful.
It is. Thank you.
Yeah, I love that so what question as a dry. I mean, my advisor asked the same thing, and we're in totally different fields, but like, so what? Why should we care? Why does it matter? But also crappy first drafts. Yes.
Yeah, I'm all about that. Yeah. And Jay Lakshmi, you were perfect on your response. That was great, with writing the titles. So is that helpful for you? Do you have problems? Because I think you have your own in-house title whisperer over here in Jay Lakshmi.
Yeah, you can call me Jaya. That's
okay, Jaya,
easy for many people here. So yeah, I first like to write the main idea a little bit so that I can get the title out of it. Because if I straight away think of writing the title and then going for writing the material, then I get stuck, and my title is never that good if I write it first. So I need to get the main idea written down in 2 or 3 sentences, and then I try to figure out the title. But I still have trouble when I try to write the title for grant applications or something like that. Because as a medicinal chemist, there are standard forms of writing a title when we are writing a paper. I try to deviate a little bit from that, but too much of deviation is also not very good, because we still want to focus on the main point. But when we are writing the grants, I sometimes struggle with writing the title for a grant, because that's something that's going to first catch the eye of the reviewers. And if we are not able to explain the exclusivity of our grant work in that title, then I feel that we are not catching the reviewer's eyes. That's something,
and that's really tricky, right? So I've worked with the scientists on their grants as well, but I think if you can, here's the key: like if you have those elements of the 4 to 6 elements of the protagonist, antagonist, conflict, scene, resolution, stakes, maybe don't put all 6. That's not going to fit in the grant because it's not a paper, right? And so you're not going to be able to fit all that in there. But maybe your stakes should be in that grant title, right? And so they understand like what's the importance of it. If not, if they're funding, if it's government or even nonprofit in the nonprofit sector, they're going to want to know why they're funding you. Just like what Tom says, so like, why should I care? And then, I like to put the protagonist in there. So what I do, sometimes I do like distill it down to like 3 to 4 of the elements and then try to tell that story. Like in the paper that using magnetic levitation to separate crystal polymorphs. I don't think that the scene was in there because the scene wasn't in there because they could do it on the tabletop, low cost, or pharmaceutical companies could large scale it. And so they intentionally left that out. So you can intentionally leave aspects out, but it'd be important to know what happens if you leave it out. If you leave out your antagonist, they're going to say, oh, do you have the receptor? Or do you have the mechanism, right? But if you put both of those in there, that might be okay, because in that paper that was published in Nature by Dr. Haribabu Athanari, he didn't have the iKicks protagonist in the title, but it was in his abstract all along, throughout every figure, throughout it was this thread that went through the whole manuscript. So, but there's no really true way of writing the titles, and so everyone's different. And but I think overall your grant should have a story. But I think if you can put in the protagonist, I think that works. But in like Dr. Athanari's, he didn't put iKicks, because if you put iKicks, you have to explain how did you get to that name, and that you just can't do that, right? And so it makes sense why he didn't have the protagonist in the title, but he did have like fungal multi-drug resistance, which is a big thing. It's a big field, right? And so really important to helping keeping us alive and making sure that we don't die of fungal infections, right? And so so is that helpful? But I think also like medicinal chemists, every field has their own little way of doing things. But sometimes when you want to publish in a higher impact, then those guidelines don't really work for the broader audience, right? And so I think whether you're giving a talk, to a broader audience or interdisciplinary, then maybe using a narrative method could help. And the only reason we came with this narrative method is because we had biochemists, chemists, physicists, and everybody was talking a different language. Oh, you can't say cause, you got to say lead this. And, just the language was quite different. But the only way we got them to agree for the first time in 7 years was we put it into a narrative, and it was like an outside, independent way of communicating, because I think it was a human way of communicating.
It's more like what happened.
Yeah.
Well, excuse me. Go ahead.
I was just going to say, in some ways, it's who, what, when, where, how, and why,
yes.
Right? And so I don't know if this is helpful in terms of grants, but whether using that same formula, but thinking about changes in the verbs Right, because you're not saying definitively in a grant that a thing is going to happen in the way you want it to, but you're exploring the possible impacts, right? So you're changing kind of the verb structure, but following that same formula. I don't know. I mean, again, this is out my depth, but this has really started me thinking about, kind of that really nice structure and formula that provides.
Yeah, but the who, what, when,
because
where,
in
why.
a paper, when you're writing a paper, we already have the results. And
Right.
we are basing everything on the results. But when we are writing a grant, it's all a proposal. So we don't have anything concrete except for the preliminary results. And we are extrapolating that entire work based on the preliminary results. So when we write the title, it can be also called as a, you are over exaggerating your expected results, or you're underplaying it. How to really capture the essence of
it's
what
an advertisement. It's an advertisement. And they say that on every grant, I went to something a number of years ago, and they said, look, your average grant reviewer makes a decision about that grant in the first 2 minutes.
Yes.
And because it's this psychological process. They read the title and they go, I don't know what the hell they're talking about here. So let's read the summary. And they read the summary and they're like, I still don't know what the hell they're talking about. Okay, that's it. It's over. That the grant's dead. And so I think in a manuscript, there's more motivation. People are there, if they're really interested in that field, they're willing to dig in a little deeper to get some new information. But on a grant, it's got to be sold right away. And, you don't want to start out and say, well, the cure of cancer is this, and because they're going to, that's obviously outrageous. But I think it needs to be laid out really fast what you're doing.
Yeah, and I think the who, what, where, when, and why. I think that's really key. I used that in an article I wrote for the biomedical science career program, and just on how to advise students to write, to ask for letters of recommendation, or how to write letters. And so we use that method. So yeah, I think that's key, because they need to get it within like 5 seconds. If not, they have so many grants. If you put yourselves in the issue, they have so many grants. Who doesn't like a good story, whether it's your grant, anywhere else, and then you want them to understand your work as well. So, but I think if they see the elements, like say if you watch a movie on Netflix and you can see, oh, that's the protagonist, antagonist, it takes the fun out of it. It messes up the magic, right? So the key is to do it in a way intentionally, but it's not so overtly. Right. And so like when you mentioned that you try to oversell, that's really wrong. But at the other part, if you don't use the narrative method, if you use the narrative method for overselling, that's wrong. But if you don't use the narrative method, then you're probably underselling your results. And that's not going to help you with your grants, your publications. So you got to hit that right balance. And that's why I like the 6 elements, because you name exactly what you have. And if it's not in there, like I use the word start code and recognition, I needed 3 more years. It was already year 5 of my postdoc. I needed 2 more years just to put the 3 words, start code and recognition. Those 3 words took about 2.5 years of research, just for 3 words. If not, I wouldn't be able to include it, right? And so I think being honest of what you have and your results are there. So extracting that in those elements just makes it a little bit easier. And if it's hard to tell that story, maybe you're missing the antagonist. Maybe you're missing, and that's okay if you're missing the antagonist. Your paper maybe not going to Cell Science Nature, but there's a journal you can submit it, and that's okay, because sometimes you don't have that. Somebody else might find that receptor, right? And so that's okay. So I think being honest to what you have is key. So
also for a grant, somebody once told me, they said, well, this is just another grant about environmental estrogens. And so it hit me. They don't understand what's unique about what I'm doing.
Yeah.
And like Jaya, you're working on these kinase inhibitors. So it's got to be the unique aspect brought up immediately, perhaps even in the title.
Yeah. And then in order to do that, it would be the scene. what's the actual scene of it, right? Why is that important? Why are these kinases important, right? Because there are so many kinases, but are these, and the way you're addressing them, why this important, right? So it's that scene, that specificity that really helps to say, oh, now I get why it's important, right? So, and if they had that receptor, that paper would have been published Cell Science. It would have been clear, because it would have been really, and if the results fit and support that the non-ribosomal peptides is leading to enterotoxicity, that would have been a huge paper, right? But they didn't have that, right? And so, but yeah, that's.
What is your opinion on a short title and a longer one? Because I see some titles that are almost 3 lines.
Yeah, I don't like those. I don't like and I've done that before.
Seem to be.
Some of my title, because I teach the method, and then sometimes they'll go off and write a title. I'm like, whoa, that's a long title, but it got through the review process. So I'm like, God bless you. You went through, and it's okay, right? But I think the longer the title, the less people are going to read that. They're probably going to focus on the abstract. Yeah. And I think that's a missed opportunity. You want an efficient title, not too long and definitely not too short. But if you have the elements, I try to do 1-word elements, but sometimes like the scene was star codon recognition, the stakes of star codon recognition. But those two words, those 2 elements were able to put in 3 words, right? And so that was efficient. But maybe I would leave out the antagonist in the title, but I would have it in my experiments to make sure that shows up, or leave out the protagonist, how they did in that Nature paper. So I learned from that Nature paper. He left out the protagonist and the antagonist, and he just had the scene and the stakes. But that's a great question.
Interesting.
I had a question, and I don't know. I'm curious if you think historians are really good at talking about their methodology. Obviously, in science, you have to talk about your methodology. It's critical. Do you think that this narrative method helps you to foreground the research process and makes it easier to talk about it? Because you're saying, that's part of the story of telling the story of the experiment.
Now, can you say that again? I'm trying to get the context.
So it seems to me that this narrative approach to writing up scientific research foregrounds the methods, right? Like, because that's part of the story, that's part of the narrative of what happens, that experiment. So does that help you when you're, with that method section, with that part of by thinking about it as a story?
Absolutely. We started going from, we have this whiteboard where we write all the experiments when I first joined the lab at Harvard Med. But we got so good in the lab doing narratives with storytelling. We would do the same thing on the whiteboard. Okay, here's the protagonist, here's the antagonist, here's the experiments. And we would actually use that to do experimental design. Absolutely. It let us know, and that's how I ended up on that paper with the 4EGI1 with that EI4E, which you correctly labeled as the protagonist. They needed to mutate all the different faces. It's like a cube. All the faces of that had to be mutated to make sure we had the right binding face, right? And what worked. And so we used the method. Was like, oh, our validation is skinny. We only had 2 mutations, so we ended up doing like 40. And then that really strengthened the validation. Let us know, no, we had the right binding interface. Absolutely. But sometimes you say, oh, we got the crystal structure, something big, but we don't have the experiments leading up to that. We might need a purified protein, see where it binds, in the cell, some fluorescent studies. We might need to see the localization of where those interactions bind. Yeah, so it's almost at every single point that it could help with.
That's amazing.
Yeah, I've seen all different derivations of use of the narrative arc, and it works really well. Just like in storytelling, like you can map a narrative arc in a lot of narratives, and I can see it in movies, and with me and my daughter, like my dad, I used to watch a lot of movies. It's in my book, and I learned a lot of storytelling from him. We try to predict what happens. So I'm the worst person to watch a movie with, because I'm the one predicting, oh, it's this, it's that. And so now me and my daughter, we have this game, because she's going to Caltech in the fall. I think if she accepts it, I don't know if she has yet, but she has until May 3rd. take prep. And if she goes to Caltech, I want her to be, yeah, she's wonderful at science, calculus, chemistry, physics. She got all that math. But I'm like, can you tell a story? If you can't tell a story, then like you're gonna have a hard time with these other scientists. And then, but if you can, maybe your dad might rub on. She got her smarts from her mom's side, but the storytelling, that's my job. I'm got to teach her. I want her when she goes to college, she's gonna be able to tell the story. So whether it's job interview, her classes, and learning material, but like, yeah, that's something we do, and yeah, it's I think it's really important to be able to identify, being able to see that in other stories. And then maybe I hope I didn't ruin any movies for you when you start looking. Oh, that's the protagonist. It's like, no, don't be like me. It's a horrible obsession. Like, I am no fun to watch a movie with. Only my daughter enjoys it, but nobody else likes it. It's like, can you please stop screaming out stuff? And I'm not always right, but a lot of times, I'm pretty good.
All right. So any other comments here? And it was sort of neat, actually, to see, Rafael, that you were meeting up with Amy and Thomas again for the second time.
Oh, yes.
So we're going to have to put you on the faculty as an adjunct, that you come in once a year for your special course.
I'll be happy to. And I would like to come. I need to fly down there. No Zoom stuff. I want some of those oysters again. Those were amazing.
But you know what's funny is on Zoom, it was actually neat to get the chat.
Oh, that's true.
Where in real person, we might be looking at you, but everyone's looking away going, I'm not going to say anything.
Yeah. Yeah, the chat was a little easier, right? Because you're absolutely right. It's harder. Yes, that might be. So maybe we could do a combined, because maybe not everyone's going to be able to be there. So you could do a Zoom and.
Well, I'm ready to get rid of Zoom as well, but it's just an observation I made. Is of course, students, what they do is they just don't chat and they tune out, and we don't know what they're doing.
Yeah.
Yeah, I'd also love to think about how we can. I think a lot of people saw scientific and think, bench or lab science, and I think what you have here is something that is totally applicable across disciplines. And I'd love to think about a way to kind of, marketing is not my favorite word, but really, saying, hey, you're you in education, or you in, it's all about this narrative, and finding a way to sort of make it even more interdisciplinary, and not just me being the only one that doesn't know the science over here.
Yeah, no. Well, you nailed that protagonist. So you're the only one that did that. So,
dumb luck to set it up.
Yeah.
I'd been through this before, and I was trying to do the obvious one.
No, you did great. You did great. and awesome. But there's always a split. It's never 100% the protagonist, or they always split it. And so I felt bad the first times when they were saying it wrong. So then I was like, okay, you're thinking like a Harvard faculty member. So that, and then the audience would normally laugh, and they tend to be okay with it. So you don't have to think, agree with me. So
yeah. Okay. All right. Well, that sounds good. All right. Well, thank you so much, Rafael. And I'm not sure when you're going to be in Louisiana next. I know Gus says you come down now and again too.
Yeah, no, I think we need to. He's going to work that, and also Isaiah Warner at LSU as well. So, but we'll see how it goes. Hopefully, we get out of this pandemic soon. It's been a long year. So, and I'm used to traveling, so this is to be like homebound is quite difficult. So.
I bet it is for you. Yeah, and I bet you've got a lot going on there with your job, with the dealing with all these students and their issues.
Yeah, no, that's not easy. Yeah. But I love working with students. It's rewarding though, because you really can try to help somebody, and it might not be small. It may be small to me, but to them it's really big.
Right.
So getting a W or deciding a week, it's it's really helpful to them. So.
Yeah. Okay. Well, you have a good afternoon over there in Boston, and if you guys need to get a hold of Dr. Luna, I'm sure you can contact him, and he can include you in his next seminar as some crazy public health paper that he had to change the title, or a medicinal chemistry paper. I don't know if he'd go so far as to include one of Beth's history
would.
chapters.
I would. It depends on the audience. I would. I'm open. I would love to have your pictures in the Hall of Fame if you use the method. So. All right. So thank you guys so much. And Tom, if I get that recording, I'd be great just to see how, you know.
Yeah, I would also like to get that.
Yeah,
Elizabeth will get that set up.
Because I would like to critique myself. I'm like, did you do that right? Did you do that? Oh, no. You know, so I love to kind of critique myself. So
well, it was great. I thought it was great. Thank you.
Oh, thank you, Beth. Thank you, Amy and Jaya.
Well, I'm glad you liked it, Beth, because I was worried that bringing in the biochemist for the history major was going to be rough.
I survived. I did it.
Yes, you did. All right, you guys take care now.
Okay.
All right. Thank you.
All right.
Bye-bye.
Bye.
All right. All right.